In the last lecture, we got to some pretty complicated stuff, and I'd like to start this lecture with a review of a dihybrid cross. Remember that a dihybrid cross is a genetic cross that tracks the inheritance of two different traits. We used the example Mendel's peas, and the two traits in question was whether the pea was round and whether it was yellow or whether it was wrinkled or green. Okay? So we had our two pure breeding parental lines, and we crossed them, and 100% of the F1 were round and yellow. Okay, that's the F1. If we take two of the F1 and cross them, we get an F2 generation that has four possible combinations of these two different traits. Round and yellow, which was present both in the parental and in the F1 generation, but then new ones. So round with green, which we didn't see in the parental generation. Wrinkled yellow, we didn't see that either. And then wrinkled green. We did see that back at the beginning as well. The thing is that these follow a particular ratio. So the empirical observations here are 315 to 100 to 100 to 30. And that works out roughly to be about 9 to 3 to 3 to 1. 9 of the double dominant phenotypes, 3 with one dominant trait recessive for the other, and vice versa, another 3, and then the last one, only one double recessive trait. Okay, we explain this through the law of independent assortment, which allows us to understand the pattern of inheritance in a dihybrid cross. We have our diploid genotype producing haploid gametes, and in our double heterozygote, which we'd have in our F1, that last example, the four different genotypes of the gametes are equally likely. So one that has both the dominant R and dominant Y alleles in that gamete, another that just the capital R and the little y, vice versa, and then one with the double recessive. Okay? So the capital R is just as likely to be found in the gametes with a capital Y as with a little y, and likewise the capital Y is just as likely to be seen with big R as little r. Okay, so these are just as likely to be found, all four combinations. Meiosis is essential for understanding this process because if we had our dominant alleles coming from the same purebred ancestor and our recessive alleles coming from the other purebred ancestor, there's nothing, in fact, that keeps those together when this individual now produces gametes. Yes, sometimes the capital A will be in with the capital B again in the gamete, but it's just as likely that the capital A will be with a little b and vice versa. So these are being passed on independently of the parental generation's combination. So if we look at our pedigree again, our round yellow is pure breeding, so it's double homozygote, and our wrinkled green is also pure breeding, so it's double recessive. We cross them, and all the F1 generation is double heterozygote. It's heterozygote for the round wrinkled locus or gene, and it's heterozygote for the yellow gene. Okay? So we take two of these double heterozygotes, cross them, and we get four possible combinations of the gametes of one parent, four possible combinations in the other parent here, these double heterozygotes, and then we get 16 combinations in the offspring, in the zygotes produced by these gametes and these gametes. So 16 possible combinations in the children, and we saw before that there's all these different genotypes if we really knew the exact genotype. But in Mendel's times and in a lot of breeding experiments, we don't know it, we just know the phenotype. And so if we track the phenotype ratios, there's 9 ways that we expect to get at least one capital R allele in the same individual as at least one capital Y allele. So there's those 9 right there. And there's just one way that we would have had the double homozygote in the offspring. They had to have the double homozygote, they had to have the double recessive from this parent and the double recessive from this one. So there's only one way to get the double recessive and thus homozygote recessive phenotype and genotype. And then there's 3 different ways to have one copy of the capital R with no capital Y's, and 3 different ways to have capital Y with no capital R's. So there's 9 to 3 to 3 to 1. Phew, okay? That's tedious, and it's painful to go through, but we need this in order to understand fully about Mendelian genetics. So I now want you to go through the same painful procedure with an example here. Imagine you've got a breeding experiment in rabbits, okay? And there's a couple of traits that I want you to keep track of here. So we have a spotted rabbit. That means its coat color is spotted, and the spotted trait is dominant to solid, which is little s, and the black coloration is dominant to brown, okay? So capital B is dominant to little b, okay? So here's what I want you to do. Please get out a piece of paper and figure that we've got a brown spotted rabbit from a pure line is mated to a solid black, also from a pure line. What are the genotypes of these 2 parents? Okay? Take a couple of seconds here and say what you think is the right answer of these different possible genotypes. And what we're really asking is, what is the genotype of a brown spotted rabbit from a pure line? And what is the genotype of a pure, solid, black lineage? Okay? Okay, what did you think? To be a brown spotted rabbit, you have to have 2 copies of the brown, the little b allele, and spotted is capital S, so that pure breeding line is little double b and capital S, okay? And then we have a solid black, so black is capital B here, okay? And little s is solid. That's a recessive, okay? So if you put down C, that's right, okay? Now, next question. What would be the genotype and phenotype of an F1 offspring? Now you can stop the tape now to give yourself more time, or I'll go ahead and tell you. All of them would now be heterozygote at both loci. They'd be double heterozygotes. So all the F1 offspring would be black and spotted. Now, what if we took 2 of these F1s that are double heterozygotes and we crossed them? What would be the expected phenotypes of the F2? Now, we've known from past examples that if we cross 2 double heterozygotes and we have this kind of Mendelian genetics with complete dominance, there's no way we'd get A, right? Because they couldn't all be black and spotted. And I don't think we're going to get that ratio of half black and half spotted. We've seen that ratio of 9 to 3 to 3 to 1 before, okay? And so really, it's whether you remembered which is the dominant and which is the recessive, because one of these is double dominant, and that'll be 9 of them, and one is double recessive, and that'll be the other 1. So if you chose C, you'd be right. 9 black spotted, because spotted is dominant, black is dominant, okay, that's a double dominant. 3 black solid, so it's got 1 dominant trait and one recessive trait. There's only 3 of them. Another 3 with the traits reversed, recessive in color, but dominant in whether it's spotted or solid. And then only 1 double recessive. So if you answered C, well done. Okay, to see this in this sort of same Punnett square that we had before, we have our brown spotted. So it's homozygotes little b with homozygotes capital S to get the brown spotted phenotype and to be a pure breeding lineage. And then black solid, it had to have 2 copies of the capital B and 2 little s for be a pure breeding line. This produces all double heterozygotes, so capital B, capital S, little s. We cross them. The combination of alleles, capital B is just as likely to be with capital S as with little b with capital S as with little s. And likewise for the other parent, then we get, there we are, 9 ways they're going to be spotted in black, 3 ways they're going to be black in solid, 3 ways they're going to be spotted in brown, and just 1 way they're going to be brown and solid. 9 to 3 to 3 to 1. That's our phenotype ratio, and that's what we'd always expect to see when we have 2 traits, independent assortment. Now let's just go through one more example. So it's a dihybrid cross, so we're going to have 2 different traits, and one is height and the other is coloration. This is going to be a Snapdragon example. So tall, capital T, is completely dominant to dwarf, little t, while red flowers are dominant, capital R, white is recessive, little r, and the heterozygotes are pink, okay? So we have a dwarf red Snapdragon. We're going to cross it with a true breeding white tall Snapdragon, okay? What are the genotype and phenotype of the F1 individuals? Okay, so we've got a dwarf red Snapdragon is crossed with a true breeding white tall Snapdragon. Okay, so we know we've got homozygotes at all of these different loci. So our F1 is going to consist of a lot of heterozygotes. Okay, but because of incomplete dominance now, we're going to have a tall offspring, because tall is completely dominant to dwarf, so that's still complete dominance. But now they're going to be pink because there's incomplete dominance.